some circles, but he's never been here before, so I'm going to introduce him. He actually taught me virology. That's why he has gray hair. I'd like to introduce you to Vikram Misra.
Okay. And there's a chain here somewhere, right? Oh, here it is. Right. So, first of all, I'd like to thank the organizers, Helen and Aaron and all that, for inviting me to talk to you about something that I find absolutely fascinating, and they are these guys. Bats are really quite amazing. They're the only mammals that have acquired the ability to fly. And they did this about 50 to 70 million years ago. Now, for those of you who, like me, have tried to get off the ground by flapping your arms really fast, would have realized that in order to do that, in order to successfully get off the ground, you have to use an enormous amount of energy. And what happens is that when you use a lot of energy is that it has consequences. You have oxygen-free radicals, your temperature might not be able to, might go too high, and so on. And so bats have adapted themselves over these millions of years to deal with all of that. And these adaptations have had some other consequences, and that is that they have this wonderful relationship with their viruses. They don't seem to get sick. Some bat species have even discovered the fountain of youth. They don't seem to get old the way we do. And at least anecdotally, we know that they don't seem to have this kind of neoplasias that other mammalian species have as well. Now, the mechanisms of all of this, we really don't understand all that well. All right? The other thing about bats is that they seem to harbor an awful lot of viruses. And over the last 30 or so years, several of these viruses have spilled over into people and other species, causing some fairly serious disease, often fatal disease. Now, for some of these viruses, like the, oops, what happened? Oh, right. For some of these viruses, like Nipah virus in South Asia, Hendra virus in East Australia, and Marburg in Africa, the evidence is really quite strong. So you can find these viruses in the bats, and you can trace their transmission to humans and other species. For other viruses, like Ebola in Africa, and all the coronaviruses, like the Middle Eastern Respiratory Syndrome, severe acute respiratory syndrome, the porcine epidemic diarrhea, and severe acute diarrhea in pigs as well, the evidence is largely circumstantial, as it is with Ebola as well. And their evidence is this, that you can detect viruses that look like these viruses in bats, but you can't actually find the actual culprits. So it's largely circumstantial. Now, I want to make a couple of points over here. Firstly, none of these viruses that come from bats, or viruses that look like these viruses, actually cause disease in their primary species. There's no pathology, as far as we know. In fact, you can take Ebola virus and put it into bats, even though we're not sure about that, and you see absolutely no disease. The other thing is that although we know an enormous amount of stuff about some of these viruses, most of that information comes from what they do to people or domestic animals or laboratory animals. There's relatively little data, apart from some work on Nipah and Hendra, on what these viruses do or what their relationship is to the species within which they have co-evolved. And that's kind of important, because if you're going to find out why these viruses don't cause any harm, then you really need to know what this evolved relationship actually is. All right, so what I'm going to do is to tell you a little bit of a snapshot of the viruses that have spilled over, causing outbreaks. And then I'm going to tell you about what we're doing to try to establish what this relationship actually is and what happens when this relationship changes. Okay, so the story really begins with Marburg virus in 1967, when people in Marburg in Germany, Frankfurt and Belgrade started to become sick with a hemorrhagic disease. And soon it was realized that all 3 cities had laboratories for a pharmaceutical company, and they were soon able to isolate a virus that looks a lot like the Ebola virus from tissue culture cells derived from a primate. And once that was solved, our Marburg in our consciousness, in Western consciousness, kind of went away. The virus didn't go away. It kept bubbling up in Central Africa every few years, but in the West, we don't really care all that much about what happens over there till, by the way, even though the virus was discovered in 1967, its relationship to bats was not really established until about 2008. So we didn't really know it was a bat virus until then. So what happened in 2008, however, is that 2 women became sick with sort of a mysterious disease, one in the Netherlands and one in Colorado. And it turned out that both of them had Marburg. The woman in the Netherlands actually died of it. So what was common about these 2 women? They had both visited a cave called the Python Cave in Queen Elizabeth National Park. And the reason why it's called the Python Cave is because on the ground you see these large snakes slithering around, and they're a big tourist attraction. But if you kind of look above, you find why the snakes are so healthy and well fed. And because they're living on a smorgasbord, really. So they just reach up and eat these things. Of course, the cave has now been closed, and a lot of the bats actually carry Marburg virus. Ebola. Again, circumstantial evidence that bats were involved, and case number 0 was probably playing in this tree where there were several bats that are thought to carry Ebola-like viruses. Then you have Hendra virus in Australia, and again, these are fairly new stories from fairly recently, and it happens every year. The virus goes from bats into horses into people, and you've lost a couple of veterinarians to that, and now veterinarians in East Australia are really kind of cautious about attending sick horses unless the horses have been vaccinated. There's a terrific vaccine out there, and so if people vaccinated their horses, they could stop this chain, but there's a lot of resistance to vaccination, and so the problem continues. Nipah virus started off in Malaysia, bats to pigs and pigs to people. In Malaysia, they killed off their pig industry and kind of solved the problem. It appears again in Bangladesh, and it again is a constant problem every year. Now this news story is from this year, again an outbreak of Nipah virus. Deadly disease, it has mortality rates anything from about 30% to about 80 or 90%. So chances are you get Nipah virus, you're probably not going to survive. So what happens in Bangladesh? Pigs are not involved. What happens is largely there. They have palms, sugar palms, and people collect sap from these things. And the bats, of course, they have nowhere else to go because their natural fruit trees in the forests are no longer there. So they go and feed on the sap, fall into the sap, which is then sold. And so that's Nipah virus. Of course, we have Sars that appeared in about 2002, caused a pandemic. Canada was particularly affected by it. They shut Toronto down, which may or may not be a good thing, but anyway. But then it disappeared. However, it was, again, the reason why it appeared was you've got bats that carry this virus or virus very similar to that. They then ended up in a very stressful situation in wet markets. The virus went from bats into civet cats, and civet cats, it evolved in civet cats and then went into people who work in the markets and then just exploded around the world. There's another virus, very much a problem nowadays. It's called the Middle Eastern Respiratory Syndrome, again, very similar to SARS. And again, recent outbreaks, every year you have an outbreak. It seems to have gone from bats into camels and camels into people. So what's common about all of these viruses is there are two themes kind of run through all of these spillovers. One is that we've changed the situation in such a way, and we humans, so that bats are now coming in contact with species of animals, us and other animals, that they didn't actually interact with before. For whatever reason, their food supplies are gone, or we have now started encroaching into their territories. The other thing that runs through all of this is that in all of these situations, bats are stressed. And when they're stressed, we think that's when these spillovers occur. All right, so what we really want to do is to test this broad hypothesis. And that is that we think bats have a wonderfully balanced relationship with their viruses. They don't get rid of the viruses, and the viruses don't make them sick. The viruses just sort of carry on as long-term, low-level persistent infection in these animals. And the reasons for this are host factors, obviously, the bats, the viral factors, and of course, environmental factors as well, that keep this relationship in a balance. If you disrupt this relationship, then what happens is the viruses then get the upper hand, they start to grow, and that's when spillovers actually occur. So this is what we want to test. And how do we want to do this is this. Again, I told you that it's important that we learn what these viruses are doing in the species that they have evolved in. So what we are working on are two of our local species, little brown bats and big brown bats. Here are their ranges. And we've isolated two different viruses from these bats. The little brown bat coronavirus, we haven't isolated it, we can detect it, but we're trying to isolate it, and a big brown bat herpes virus. And we've got a cell culture model of big brown bat cells, and so we can sort of do a lot of experiments in the cells trying to determine what's going on. And then we also then look to see what actually happens in the bats as well, to see whether answers that we get here actually apply in real life or not. And then answers that we find here, we can then go back to our cell culture model to see whether we can tease out what's happening at the molecular level. So we've isolated bat gamma herpes virus. What we find is about 70 to almost 90% of bats that we look at are infected with this virus. And as you all know, herpes viruses, you get it for life. The virus stays latent, and it's reactivated in response to stress. So we're hopefully going to be able to do that. And then we have the bat coronavirus. We have the whole sequence of the virus. It looks very much like the porcine epidemic diarrhea virus, and is also fairly closely related to the human H229 virus, which is another coronavirus. It's a little more distantly related to the Sars and MERS viruses. So the question that we asked is, what is this wonderful relationship between bats and their viruses? Or why don't bats get sick? Now, before we answer that, why do we get sick when we have a viral infection? And why are some viral infections that much more severe than others are? It's a really complicated question. There are probably all sorts of reasons why we get sick and bats don't. But one thing that's important is inflammation. Now, when we are infected with a virus, two things happen. Our cells make antiviral proteins called interferons, which suppress viral replication and do all sorts of wonderful things. We also turn on our inflammatory response. And when the inflammatory response and the antiviral response is balanced, it's a good thing. It all aids in recovery. However, when the balance tips and there's too much inflammation, then we start having problems. And this is an issue with severe cases of influenza or any of the coronaviruses or any of the viruses which cause severe viral diseases. You have way too much inflammation than you actually need. All right, so we decided to see what happens when you compare bat cells and human cells. And Arinjay, who's a PhD student in the lab, took bat cells and human cells, and he stimulated these cells with a molecule that resembles viral nucleic acid. And the reason why he used this surrogate of viral RNA was to see what the cells were actually capable of. And the reason for that is that all viruses have a way of shutting down some of these responses. And so he wanted to see what they were capable of. So he did this. And so what you find is if you look at interferon, both human cells and bat cells respond really in a robust way to this molecule. If, on the other hand, you look at TNF alpha, which is a molecule that turns on inflammation, you find a huge difference. One is that human cells respond in a very robust way. Bat cells, there's absolutely no response at all. It's not that bat cells don't have TNF alpha, they just don't respond. In fact, they have a very active mechanism for turning off the interferon response. So now what happens with viruses? Now, I told you that we haven't isolated the bat coronaviruses yet, so we looked at the next best things, which is the Middle Eastern Respiratory Syndrome virus, a virus that is thought to have come from bats into camels and into people. Again, if you look at amount of virus produced, lots of viruses made in human cells, and then the human cells all die out. The virus just essentially kills them. If you look at the interferon response, you find that in human cells, there is absolutely no interferon response. And that is because the virus can turn off the interferon response. Look what happens in bat cells. The virus turns on the interferon response, the antiviral response, but the virus is not able to turn it off. So there's something very different about bats where viruses are incapable of turning off the interferon response. If you look at the pro-inflammatory response, just the opposite situation. Nothing in bats, lots in human viruses. Human cells. All right, so what exactly happens to the bat cells? Human cells die out. Bat cells, on the other hand, if you look at the amount of virus being produced over days, it jumps up and down, and then it sort of settles on into a low-level infection. The cells keep producing virus. They don't die. You can keep these cells going for months at a time. This simply shows that all the cells are infected, and they keep going. Now, one of the reasons could be, the reason why the virus persists is that the virus is not able to turn off the interferon response. So if you artificially turn off the interferon response in these things, viral replication goes up. And if you turn off some of the pathways of interferon that lead to interferon, again, you have an increase in replication. So bat cells are unique in the sense that they respond to viral infections in different ways because they don't have, they retain their antiviral response and don't have an inflammatory response. And we're trying to figure out what that happens. Now, what happens to the virus in these cells? And this is really interesting. So here you see the genome of the MERS coronavirus. This is all sorts of genes over here. Several of these gene products will turn off the interferon response. Right? Several of these genes are what we call accessory genes. The virus doesn't need them to replicate, but in the animal, they're important because they sort of turn off the animal's ability to fight the virus. We looked at what happens in the bat cells over a period of time. And each of these figures represents one of the virus's genes. This red line is what you see in persistently infected cells. Green line is what you see in an acute infection. All right? In each and every one of these cases, you have expression of these genes throughout the period of time, except for one of these genes, which we call open reading frame 5. And this is one of those accessory genes that the virus needs in order to multiply in animals. So what happens is to this gene? Well, very soon after these viruses infect bat cells, a variant of the virus is selected in which this gene is deleted. Now, we don't know what the selective pressures are for this. We've repeated this experiment, and either this gene is deleted within about 2 or 3 days of infection of bat cells, or you start to select for mutants in which there is a truncated protein. And now we're trying to sort of see what these genes actually are and how they are, what is so different about bat cells that a virus that's virulent in people, when you put it into bat cells, it very, very quickly selects for these mutants. All right, so the other thing how much time do I have? 5 minutes? All right, so the other thing I pointed out was one thing that's common to all these spillovers is a stressful event. And our hypothesis is that this wonderful balanced relationship can be influenced by stress, which then, so what is a stressful relationship? So a few years ago, about 10 years ago, a very stressful thing started to happen to bats in North America. It started off by people observing dead bats around a cave in New York, and then the virus, the disease has spread rapidly westward and eastward. And this year, it finally reached some of our test caves in Manitoba. It jumped a few years ago to Washington State, probably by someone accidentally transmitting the fungal spores to a cave over there. When this disease first hit, people were able to isolate a fungus, and the idea was that the fungus was doing this. At that point in time, we weren't quite sure if it was the fungus, and so Saskatchewan happened to be one of the few places we could do these experiments, and we collaborated with several people, including U.S. Fish and Wildlife, and established that the fungus was indeed causing this thing. But at the end of the experiment, we archived everything from these bats. We archived each and every tissue in RNA later, frozen, formalin fixed, and everything else. And this allowed us to go back to see what these tissues were doing. And we find that in about 30% of the bats that we look at, either from the experiments or in the wild, we find that you can detect a coronavirus in their intestines. The level of viral RNA in these bats is very, very low. And they all have about the same amount of virus. If you look at bats that are infected with the fungus, on the other hand, you see quite a different picture. You find that there is a huge difference in how much fungus how much virus you have in these bats. Some bats have very, very low levels, but some bats have extremely high levels of fungus. So as I said, at the end of the experiments that we did with the fungus, everything was archived, and a couple of pathologists in a blinded way had gone through all the bats and classified the lesions. And so we had all this information. We went back, and what we find, though, is that there is a direct correlation between the amount of virus that you find in these bats and the amount or the level of pathology that the fungus produces. So, we thought that, well, whatever the fungus is doing in the wings is somehow affecting what happens in the intestines. So we went back and we tried to sort of see what genes were being expressed in the intestines of these of fungal infected bats and not infected bats. And what we find is that there are pathways that that all are aimed towards turning off the interferon response. And so then we went back to our cell culture model and showed that this was indeed true. If you turn off these pathways, the level of interferons goes down and the level of virus goes up. So as a result of this thing, we came up with a hypothesis that normally you have low levels of coronavirus replication in the intestines. When you have a fungal infection, you influence all of these pathways somehow, and we're trying to figure out how a skin infection influences what happens in the guts. And as a result of that, you have massive amounts of viral replication, which then presumably would increase the amount of virus being shed and increase the chances of spillovers. Whoops. Again. I'm worried about, we have this Mac and PC issue, and I sent over a PDF file hoping that wouldn't happen, but obviously it didn't work out. Anyway, just to kind of summarize, we think that bats are very unique in their relationship with viruses because over the years, over the millions of years that they have co-evolved, they have developed ways that allow this relationship to be balanced and not mutually harmful for either the virus or the host. And when that relationship changes, we think one of the things is stress. And I didn't tell you anything about our herpes virus work, but that points in that direction as well. You change that relationship, and that puts one of those jigsaw puzzles in place that lead to spillovers from these animals. We're trying to figure out what these adaptations are in bats, and hopefully, if we can, they may lead us in some ways into trying to understand why we are so different from bats, other than the fact that we can't take off when we flap our wings. Anyway, so thanks so much. I don't know. Any time at all?
I think we've got time for a question or two. Has anyone got a question for Vikram? I'd better bring this over.
Thank you. It was a really fascinating presentation. I'm wondering, you said that when you passage the virulent virus through bat cell culture, the virus mutates to become less virulent. If you take this less virulent virus and put it back into human culture, does it regain its virulence?
That's a terrific question. So what we're doing now, what I'm showing you is fairly recent data. So what we're doing now, we've grown up the mutant and we've grown up the wild-type virus, and now we're trying to see what happens when you put them into opposite things. The other thing that we're trying to do, but you keep in mind, this is MERS virus, so it's a level 3 pathogen. So all the experiments we do have to be done in a level 3 facility, which is our Intervac Vito facility. So what we're trying to do is just sort of what happens in a camelid, because MERS went presumably from a bat to camels and then into people. So now we're trying to sort of see what selective effect both human cells and camel cells have on all of this. A great question, and I would love to know what happens.
Thank you. Maybe we should talk to each other like this.
I think that's for our benefit.
Most public health guidelines around rabies prevention talk about the fact that we should be educating the population not to touch bats, particularly if they're acting abnormally or are seen flying during the day or bump into you, because they might be more likely to carry rabies. Now, based on your presentation, I would think that that's a fallacy, that bats carrying rabies are not more likely to be showing symptoms or signs. Can you talk a little bit about that? Sure.
So rabies is one exception to the rule. Okay, so rabies, as you well know, once clinical rabies, when a mammal gets clinical rabies, chances of survival are close to 0. Bats are not that different. Rabies does kill bats. It does show clinical signs in bats. However, there is mounting evidence that rabies behaves differently in bat species than it does in humans, does in other mammals. You can find, if you look at vampire bats in Central and South America, or the European lyssavirus colonies in Europe, you find fairly large numbers of animals that are seropositive for the virus. Now, what the implication of that is, does it modulate the disease? Why did they get seropositive in the first place? Did they develop clinical rabies and then had an antibody response, or was infection at such a low level or in the presence of maternal antibodies that they didn't actually get clinical rabies but developed antibodies? So all of those questions are out there. The amazing thing is that even though bats comprise 1/5 of all mammalian species, they are everywhere other than the Atlantic, Antarctic, and the Arctic. We know relatively little about bats and how they interact with their pathogens. And so all of those answers are probably, hopefully, be solved. But in answer to your question, rabies does kill bats. It is a fatal disease and they do show clinical signs. So those, the advice that you have is probably is extremely valid. Well, thank you so much. Appreciate that.
I think with that,